yourself before we get started. So today we are very excited to have Dr. David Van Valen from Caltech, and Dr. Valen received his Bachelor of Science from MIT and PhD from Caltech, MD from UCLA, before starting his lab in 2018 at Caltech. So as you can see, actually, he's a tech guy. He got all of his degrees from tech and now continuing at tech as well. And his lab has been working on a couple directions. Previously, he worked with Rob Phillips, where I think he looked at host virus interactions, and I think he's still continuing to do that in his lab. And then at Stanford, he also started doing deep learning for signal cell biology. And today we will learn more about his experiments where he applied deep learning to live cell imaging, as well as spatial omics data. And finally, I think he has another interesting direction for integrated measurements of signaling and gene expression, for which also he's using image-based data. I mean, he's been in this image-based field for a long time, I guess.
Yeah.
Right. And then he received and about to receive. I mean, I cannot see them here, but yeah. I think I'll skip that part. But without, I think, further ado, then we turn to David.
Okay. So thank you for the invitation. So today I'm going to talk about my lab's work on adapting deep learning methods for single cell biology. And so the title of the talk is Single Cell Biology in a Software 2.0 World. And hopefully in a few minutes, you'll sort of get a sense of what I mean by that. Okay, so deep learning is really changing how we're able to interpret biological images, and I think this is particularly true if you're one who's trying to be quantitative with biological imaging data. And so this slide is sort of meant to represent a wide variety of different methods that have come up in the community, some of which my lab has been fortunate to contribute to, but by no means are we like the only ones developing this method. So there's really a really vibrant community of both computational and experimental methods developers who are working around the clock to use deep learning to solve difficult challenges in biological image analysis. And so some of what is now possible is represented on this slide. So there's cell segmentation, which is going to be sort of the subject of the technical subject of today's talk. That's not all. Unfortunately, it looks like my GIFs aren't working. Yeah, which is unfortunate, but I'm just going to go through because the slide shouldn't be that long. So there's cell tracking. So if you're doing live cell imaging, not just identifying cells in every frame, but then tracking and linking them together in time. There's doing spot detection in fish experiments, and so being able to identify, where single molecules are in these large fields of view. But that's not it. So there's also some more imaginative applications of deep learning, and so two of these are highlighted down below. One of them is doing image restoration. So if you have noisy images, can you sort of restore what the denoised image would have been? And so these methods sort of, let you do experiments in regimes that weren't previously possible, just due to limitations of things like, phototoxicity and whatnot. You can image with less light, your samples live longer, and so you can see phenomena that you wouldn't have been able to see otherwise. And then there's augmented microscopy, and this is really giving new life to label-free imaging. And so here you sort of use deep learning to predict what fluorescent images might have been had you imaged them. And so if those fluorescent images contained information like where the cell nuclei, which cells are alive, which cells are dead, if the information is present in the bright field or label-free or phase images, you can sort of get that information for free without having to do the fluorescence microscopy. And so this slide, the whole point of this slide is just to say that this is a very vibrant area, and there's just a lot of very powerful methods that are either have been developed or are on their way. The reality, though, is that actually getting deep learning to work in practice requires much more than just deep learning. And that's really the main reason why the title of this talk is Software 2.0. single cell biology in the age of software 2.0 as opposed to in the age of deep learning. Software 2.0 is a term coined by Andrej Karpathy, who's the current head of AI at Tesla. And it sort of calls attention to that fact that if you want deep learning to work in practice, then you need much more than just the deep learning algorithms. And that's sort of highlighted by this figure here. So this figure sort of conceptually walks through what's required to make a modern deep learning model. And so the first part is actually creating annotated training data. So most successful deep learning methods are supervised machine learning, and so that means that they learn how to do their task from labeled data. And so if you want to create a new method, so here this is cell segmentation again. So if you want to create a method, deep learning model for doing cell segmentation, the first thing that you do isn't open up Sublime Text Editor and start writing down some code. The first thing that you do is compile a data set that actually speaks to that need. And so here, if you're doing nuclear segmentation, one would collect a collection of images of cell nuclei, and then create associated labels. So in this case, someone would have to go through these images and say this collection of pixels belong to cell number 1, these collection of pixels belong to cell number 2, these to cell number 3, and so on. Once you have that annotated training data, you can then train a deep learning model to do that task. There's a variety of, for most tasks that folks want to do, there's a variety of methods that they don't quite work out of the box, but they're not that difficult to construct and train. Once you have the training data, you can actually train models to do this task. And so then once you have a trained model and you want to use it, you then take new data, run it through your model, and then take the predictions and then go do what you would like to do with those predictions. And so this figure sort of highlights these two areas. Creating annotated training data and deployment. To actually use deep learning in practice, you need to have those two. So again, there's data, there are the models, and then there's the deployment, or you call that like the computational infrastructure. And so my lab's been working sort of at this interface of artificial intelligence and machine learning and biology and imaging for the last I'd say 2 to 3 years. And what we've realized is that to actually do this software 2.0 model well, you actually need to be co-developing data, models, and compute. It's not enough to pay attention to any one of these silos. You need to be paying attention to all of them all at the same time. And there are challenges at each one of these in each one of these spaces, which are I'd say these are the questions that sort of keep me awake at night. And so this slide doesn't necessarily say that we solved any one of them, but just to say that these are challenges for folks who are active in this space. On the data side, how much data is necessary to get sufficient accuracy to perform a given task. For a lot of what biologists want to be able to do, the labels that are required are actually the most difficult to make. And so that is for an image, you want dense pixel level annotations for entire images, how you generate them in a way that's both scalable and affordable. Biological imaging data tends to be higher dimensional. And so if you're doing live cell imaging, that's 3 dimensions, 2 in space, typically 1 in time. If you have a light sheet microscope or confocal, that can be 3 in space, 1 in time. And if you're doing the multiplexed imaging, then you can have 2 spatial dimensions, but then a number of different color dimensions as well. And then lastly, a more technical issue, data sets aren't static things, particularly if you're building data sets for machine learning. And so how to version data sets and then link them to the models and the analyses that are then derived from them. I'd say that's an open issue. On the model side, getting a sense of which model architectures are accurate enough to so that you could just automate a particular analysis task. I'd say that's still an area that's I would say unresolved. But more important than model accuracy are all the other things that make models useful in practice. So their memory footprint, inference speed, and whatnot. And so managing this trade-off between a model's accuracy and then all the other features that you'd like the model to have for it to be useful. I think that's a open problem that's not getting much attention. And then, sort of along those lines, understanding, okay, which models are actually suitable for deployment on big data. If you have large data sets, then, inference speed really starts to matter. And then, once, we get to a point where it's not just a single model that's useful, but collections of models on, figuring out like how to optimize that whole collection as opposed to optimizing individual models, that's almost certainly going to be a major challenge. And then on the computational side, integrating models into complete pipelines. Models, most successful applications of deep learning are not just deep learning, it's deep learning plus something else. And so getting these entire pipelines sort of integrated with deep learning functionality, I think that is a open, relatively open issue. Migrating these pipelines into the cloud so that they become more accessible and then also like more scalable. I think that's also an area that needs attention. And then optimizing models for use on edge devices. There are a wide variety of experiments where where the experimentalists would really benefit from real-time feedback from the models, making sure that those use cases are getting their due attention. That's also an issue. And then lastly, this isn't something that I'm going to talk about today, but it's one thing that we think about quite often in our lab is, now that we know that deep learning can do these things, if we had these issues solved, how would we update our experimental design so that we can design better, faster, more scalable experiments? Okay, and so I've split the rest of the talk into just sort of two vignettes that are at the interfaces of these three different silos. And so the first one is at the interface between data and models, and it's really about how to get single cell information on images. The second one is at the interface of models and compute, and how do you do cloud computing for software 2.0 in a sensible way. Okay, so single cell analysis is a common challenge for a wide variety of biological imaging experiments. So in our lab, we study cell signaling, and so the experiments look something like this. There's a fluorescent reporter that, where either it's the brightness of the reporter or its location informs you about what a signaling pathway is doing. And so if you were to image the activity of these reporters in different environments, you can, in a very real sense, in a very real way, get a sense of what the cell is thinking about the environment that it's in. And then we're also interested in how these dynamics are then tied to different changes in cellular phenotypes. So for instance, gene expression, cell morphology, and whatnot. And so if you're interested in changes in gene expression, you might stop your experiment at a given time point and then do an endpoint assay like single molecule fish. And so what you want to be able to do is take those movies and then quantify them. So the reporter that I showed in the previous movie, its location, whether it's in the nucleus or the cytoplasm, sort of report on that pathway's activity. What you'd want to be able to do for those movies is then segment cells in every frame, link them together over time, quantify fluorescence, and then get these nice traces for every cell, how much fluorescence was in the cytoplasm versus how much in the nucleus. For fish data, what you'd like to be able to do is identify spots in your image. If you're doing a multiplex fish experiment, then you want to be able to register those spots over the different rounds of imaging that you do. And so, computationally extracting this information is a challenge, and we're working on in my lab, we're working on solving these problems with deep learning. I'll get to that later at the end. Unfortunately, I don't have time to talk about the fish data analysis in detail, but what I will say is that broadly speaking, the big challenge for this problem is, what is the ground truth? Just because the reality is that these images usually contain, tens of thousands to hundreds of thousands to millions of spots. There's no way a human being can annotate, what's a real spot, what's a false spot. And so the solutions that folks have come up with to sort of get around this problem are sort of in two different bins. One bin is, let's simulate what the ground truth should be. And so methods like Deep Storm essentially do this. And so if you know some information about your microscope, what the point spread function is, or some approximation of it like a Gaussian, then you can simulate what you imagine your data might look like. And because in your simulation, where your spots are, you sort of have it baked in like ground truth. The second way is to sort of take more classical algorithms, run them over the images, fine-tune them, and then use the fine-tuned version as a quote-unquote ground truth. And that second version is sort of what we've been pursuing for the last couple of years. Broadly speaking, it falls into this weakly supervised category. And so what we're in the midst of doing is sort of taking all the existing classical algorithms that are out there, optimize them for a collection of images, and then use generative modeling to sort of find consensus among these different algorithms, and then use that to train a deep learning model to do spot detection. And fortunately, we're still a work in progress, but as you can see from the image, our early successes, our early data looks quite promising. Okay, the live cell imaging isn't the only space where single cell analysis is of value, and I think another area is tissue imaging, and particularly tissue imaging with new multiplexed imaging methods. So new methods let you image large panels of antibodies all at the same time. Roughly speaking, they fall into two categories. One uses iterative rounds of staining, imaging, and destaining. The other uses mass cytometry to as an imaging modality, and you sort of need to do one of the two to get around the problem of spectral overlap if you're imaging with light. Yeah, and so these multiplex imaging methods let you collect great data in tissues, and so this is an image from a paper that I was fortunate to be part of. By Liat Karen and Michelangelo, came out in Cell 2018. And so this is a multiplex ion beam experiment where we imaged a collection of triple negative breast cancer patients. And so there are different markers, double-stranded DNA, which highlights cell nuclei, keratin, beta-catenin, smooth muscle actin, CD45, EGFR, P53. There's a whole other set of markers. The whole point of this image is just to show that for different patients and different samples, you can collect a multitude of different markers. I will add there's also a transcriptomics counterpart to these methods, and so these are the multiplexed fish experiments that are moving their way into tissues. A lot of what I'm going to talk about today is relevant for those experiments as well. I felt a need to highlight those. Okay, and so these new imaging modalities produce really rich data, but at the end of the day, there are a wide variety of computational challenges, and the very start of almost every computational pipeline is single cell analysis. What you want to be able to do is to go into these images and say, okay, this set of pixels, cell number 1, cell number 2, 3, 4, 5, 6, 7, or whatnot. Once you know where the cells are, you can do tasks like assess the marker intensity for all the different markers, identify cell types, identify cellular behaviors or whatnot. Okay, and so this is actually the contribution I made to this paper, which is we sort of had like a pilot deep learning algorithm at the time, and sort of getting it adapted to perform reasonably well on MIMI data for doing nuclear segmentation. And so the segmentations were sort of good enough to go ahead and quantify, and you're able to once you have that information, you can do stuff like this. Where here, we've sort of highlighted all the different immune cell populations that were present in this patient's tissue. These sort of analyses, and particularly being quantitative with them, is not possible without accurate single cell analysis. Okay, so since then, that paper is like now a couple years old. We sort of been exploring like, okay, what's the best way to do this? And so, from the deep learning perspective, there are a wide variety of ways to identify single cells with deep learning. This slide sort of categorizes the different methods that folks in the community have come up with. Broadly speaking, they fall into 4 different categories. There's pixel classification, so trying to identify for every pixel in an image, is it inside of a cell, at the cell boundary, or part of the background. There are deep watershed methods, which try to ascertain for every pixel inside of a cell, how far is it from the cell background. And if you have that information, you can then use classical computer vision algorithms like the watershed transform to separate out neighboring cells and finish the segmentation challenge. There are methods like mask-R-CNN, which seek to encapsulate cells in different bounding boxes. There's a step called non-maximum suppression, where if there are bounding boxes that have high degree of overlap, you turn off the one that has the lowest score. And then once you have bounding boxes that sort of neatly represent single cells, you can then find okay, within that bounding box, which pixels am I going to say is that actually belong to that cell and which ones are the background. And then there are methods which are called vector embeddings, and these basically try to color assign each pixel in an image a color and do it in such a way so that different cells get different colors. And that's not sort of represented pictorially here, but these are the other sort of the last category. All the different methods that folks have come up with in the community that sort of fall into one of these one of these four methods. What we use in our lab are although we've implemented all of them, and I'd say like there actually is no silver bullet. Which method one would use depends on the type of data and its characteristics. But generally speaking, in our lab, we like these deep watershed models, just because they're as accurate as anything else that's out there, and they have some nice features for deploying on larger images. And so roughly the way these work is, you start with an image. Deep learning models actually have no idea about location in image, and so they actually aren't aware that this would be like the upper left, and this part would be the lower right. And for a deep watershed approach, which sort of does try to suss out distance and location, it is helpful if the model sort of had access to that information. So the one thing that we do, which we found gives us a mild boost in accuracy, is we append a coordinate map to the images before they go through the deep learning cycle. And so this coordinate map sort of gives the location information back to the deep learning methods. There's a price. You lose sort of the translational invariance. And so you have to sort of be relatively aware of that, for all the downstream stuff. But we have found that this helps with the training process. Then we train deep learning models just to predict two transforms. So one, it tries to predict where's the cell center of mass. And the second is it tries to predict the deep watershed transform. How far is a pixel inside of a cell to the background. And if with these two images, you can sort of use these as inputs to a marker-based watershed algorithm, and you produce some pretty nice cell masks. And we found that these work quite well for a wide variety of different image types, which sort of show later in the talk. But every deep learning segmentation model in this talk is using some derivative of this approach. Okay. So models are one thing that can be improved, but I think the reality is that the real path to improving model performance is by improving the underlying data. And this is just an observation that we found, is that models don't perform when the images that they're operating on lack the information necessary to perform the desired tasks. For live cell imaging, this means that there's a lack of temporal information in the training data. This is somewhat impactful for segmentation, but if you wanted to do cell tracking, this is a real issue. You need sort of whole movies annotated if you want to have accurate cell tracking done through a supervised machine learning approach. For multiplexed imaging, and honestly just tissue imaging in general, the reason why models don't perform is because there's a lack of contrast between neighboring cells and tissues. Because cells are in close opposition, that close opposition erases contrast in the images. And so this is sort of shown in this example here. So here we have like images of cell nuclei, and if you're to train a deep learning model to just predict the boundaries between cells that a human sort of annotated, you'll get something like this. And so you'll see, there are some, pretty striking contours, but there are all these areas of gray, which represent the deep learning model just being uncertain. Am I inside of a cell, or am I at a cell boundary? And that's just because the images themselves lack contrast. Multiplexed imaging sort of has a unique opportunity to solve this problem through data engineering. And what I mean by that is that because you have all these additional channels that you can reserve some of them for adding in structural information. And so here what we did is we included an image of the same tissue that's been stained with an antibody that targets a nuclear envelope. And these nuclear envelope image then effectively adds that contrast back. And so when you train a model on that, nuclear plus the nuclear envelope image to do the cell boundary prediction, what you get is the following. And so what you see is like all those areas that were previously confused, those those effectively go away. Yeah. Okay. And so really what this means is that to get performant models, it's not the path isn't through more deep learning engineering. The path is actually through large scale data annotation, and that's what we've been embarking on for the last 2 years. And so we've sort of been working on these two annotated image collections. The first one we're sort of calling an ImageNet for multiplex tissue imaging. And the goal here is to move beyond just nuclear segmentation. Well, nuclear segmentation has been the bedrock of most single cell analyses in tissues. The reality is that it's inadequate. Most markers aren't nuclear. And so if you're using nuclear segmentation as the basis for quantifying those markers, you're bound to be inaccurate. But on top of that, you can't do subcellular localization analyses if you're relying just on nuclear segmentation. And you also can't do morphological analyses as well, because the morphology of the whole cell is quite different from just the morphology of the nucleus. And I think the need for whole cell segmentation is particularly striking for the multiplexed FISH platforms, because again, most of the FISH spots that you want to be able to detect, those aren't nuclear. They're spread out through the entire cell. And as I mentioned in the previous slide, multiplex imaging and deep learning sort of provides a unique opportunity to solve this problem through data engineering. Building data sets where you have both nuclear images and then membrane/cytoplasm images, just making sure that you have enough information present so that you can actually do the whole cell segmentation. And I think the reality is that presently folks are actually trying to make sure like the images of this flavor are included in these experiments. Okay, so as I mentioned, we now have the opportunity to actually like have images that have the structural information to perform segmentation. And so what we've done is we've compiled nuclear and also membrane/cytoplasm images from essentially every multiplex imaging platform that's in use today. And so these are the mass cytometry methods. So imaging mass cytometry from Fluidigm, MIMI from IonPath, and then also the fluorescence-based methods as well. And so this is Codex from Akoya, multiplex immunofluorescence from GE, SciSif, which Peter Sorger's lab has pioneered, and whatnot. And so we have a large collection of images representing these different platforms across a diverse set of tissues. The real challenge is annotating this. And just to show what why this is hard, I just want to show like a variety of different images that we have that sort of representative in this data set. What you'll see is that cells vary in their morphology, both like across tissues but also within tissues. There are differences in signal to noise across different platforms, and also differences in what's autofluorescence. The mass cytometry methods, because they don't image with light, they tend to have less autofluorescence, although admittedly the images tend to be noisier. And then you'll also see that in some of the images, you actually have cells that don't have cell nuclei, and this is sort of an artifact of taking two-dimensional cross sections through three-dimensional objects. You're not necessarily guaranteed to be able to capture the cell nucleus in those cases, and so being able to perform segmentation in those cases is something that one would like to do, and it's not currently possible if you're just doing nuclear segmentation. And so to sort of get a sense of how hard this is, so again, these are the 4 challenges: variation in appearance, signal to noise, and autofluorescence, cells sort of lacking nuclei, and then also just like the dense packing. To get a sense of how hard this is, we've actually had human annotators annotate images from different represented pieces of this data set, just to get a sense of how, if we had two people annotate the same image, how well are they going to agree? And I think this problem is sort of challenging to the point where even reasonable experts are going to disagree to some degree. And so for these different tissues, what we observed is that there's some that have fairly like a I would say like low but a noticeable level of disagreement between annotators, and there are others like sort of densely packed immune tissues where it's sort of easier to distinguish what's one cell versus what's two. But this human level performance sort of gives a benchmark as far as what can we reasonably expect an algorithm to be able to do, particularly one that's developed using supervised machine learning. And sort of the inner annotator agreement is sort of the upper level of human level of what's considered a human level performance. Well, not upper level. That's sort of what we consider to be human level performance. Okay, and so to develop these data sets, we've actually that's actually both a technology and an art. And the way we do it is to use crowdsourcing, but not having crowdsourcers work on this without help, but do it in a human-in-the-loop framework to accelerate the data annotation process. And so the basic idea is that we have two different sets of workers. We have the crowd workers who have high bandwidth, but relatively low experience, and then we have the experts who have low bandwidth but immense experience. And the goal is to sort of get these two communities in combination with the deep learning models to work together to accelerate this annotation process. And so the framework that we put together sort of works as follows. And it has 3 steps. In the first step, human experts, the experts, work together to craft a pilot data set. So they annotate some amount of data, and they use that data to train a preliminary model. That in the second set, that preliminary model is then used to process new data sets, and the outputs are then sent to the crowd workers, and their job is to correct the errors the algorithm makes, as opposed to creating annotations from scratch. And so here, they might get an image where, there's an incorrect segmentation, a merge error, or whatnot. There will also be some cells where, the algorithm sort of nailed it. And so the crowd workers work with some guidance, with some instructions laid out by the experts. They work to correct these errors. The corrected versions then goes through a quality control step, and after some expert quality control, they get added to the pilot training data. Then you train a new model on this larger data set. That new model invariably ends up being more accurate because it sees more high quality training data. And then you repeat the cycle. And so then you use this new model to process more data. That gets sent out to the crowd. The crowd, again, corrects and curates. That goes through QC. You update your training data, you train a new model. And what ends up happening is that as the model gets more and more accurate, the crowd workers get faster and faster because there's just less annotation work they have to do to create good training data. And then then ultimately you enter the third phase where the model is accurate enough where it can actually see deployment in real laboratory and hopefully in the not too distant future clinical settings. Okay, so we've sort of run this loop for this compendium of data. We're calling this data set TissueNet, and ImageNet for multiplex tissue imaging. This slide sort of goes over the breakdown. And so, we've annotated both whole cells in all of these images, and then also cell nuclei in all of these images as well. And we have approximately about 1 million nuclear segmentation in this data set, and about 1.2 whole cell segmentations in this data set. And we sort of, to get a sense of like how large this is, we sort of went through the literature and counted how much annotation has the field done. And so it turns out if you look at all the data sets, including H&E images, cell culture images, and other tissue images, there are approximately around 500,000 nuclear segmentations that exist currently out there in the field. Almost no whole cell segmentations publicly available. And so we've annotated more in this data set for cell nuclei than the entire field has done previously. And for whole cell segmentation, there's no comparison out there. Looking across these different platforms, we'll see that we have around 300,000 images from Codex, similar amount for SciSif, a smaller amount for Vectra, around 200 to 250,000 for MIBI, around 100,000 for the multiplexed immunofluorescence, and around 50,000 for imaging mass cytometry. And there's a similar breakdown across different tissue types as well. So, immune cells, GI, pancreas, breast, lung, and skin. Using this data set, we can then train deep learning models to do both whole cell and nuclear segmentation. And so the resulting model we're calling Multiplexed DeepCell. I will admit that our team is not settled on a model name, and so this might actually change between this talk and when we actually get a manuscript together and out the door. But right now we're calling it Multiplexed DeepCell, and this model does the same model does nuclear segmentation and whole cell segmentation. The input is images, and so again you have a nuclear marker and then a membrane or cytoplasm marker. This goes to the model. The model predicts where the cell center mass is, and then also it predicts sort of a cell boundaries. Well, it predicts the pixel-wise transform, and then that gets well, basically one of those gets sent to produce a sort of a distance map, very much like the deep watershed approaches. And so in combination of having both the shape information and then centroid information, you're able to produce sort of predictions of where the cells are in these images. This model is, while it's useful in and of itself, it's actually most useful as part of a complete pipeline. Images that folks want to analyze are of different shapes and sizes, and there's also a need to be able to perform the pre-processing and the post-processing steps all as one package. And so we've done that, and again, to reiterate, complete solutions require more than deep learning. And so, like a software package that we put together, it'll tile images into these reasonable patches. Each patch then gets normalized appropriately. Then the inference is done, and then the outputs are then stitched back together. And then once you have it stitched back together, this goes through post-processing to produce the predictions of where all the cells are in the images. And just to reiterate, not to sound like a broken record, but complete solutions require more than just deep learning. So these models, this model, one model trained on TissueNet, generalizes across all platforms and tissues. And so here, I believe this is an earlier version of the model, but this just shows a smattering of different tissue types and platform types. So Hodgkin's lymphoma and Vectra, colon in multiplex immunofluorescence, breast cancer, ductal carcinoma in situ, immunofluorescence in MIBI, lymph node images in immunofluorescence, and then melanoma data set from MIBI. Just to sort of show like the latest version, I think I have a couple slides. And so this is an image of pancreatic ductal adenocarcinoma lent to us from Travis Hellman and Memorial Sloan Kettering. And so when you run it through the entire pipeline, these white outlines are the predictions of where the cells are in this image. The nuclear predictions are also present, but they're not shown. They're not shown here. Similar information, just visualized differently in lung cancer from a SciSif data set from Peter Sorger's lab. We benchmarked this thoroughly. We have what we think is a nice benchmarking package for cell segmentation. And here what's shown is the recall precision and then the Jacard index. Recall measures if an object is there, will the model pick it up? Precision measures if the model detected it, what's the chance that it's actually correct? And the Jacard index measures the amount of pixel overlap between of the cells that are correctly identified. How well do the actual segmentation masks agree? And so what we see is that we sort of broke it up into two different comparisons. These are models that were trained on TissueNet, and so here we have this multiplexed DeepCell. Retina mask is a sort of variant of mask-R CNN, and so you'll see is that while the precision is similar, the retina mask suffers with recall. It misses a significant number of cells. And then the feature nets sort of represent like the very first iteration of deep learning models that we developed back in like 2016. And so these comparisons are sort of meant to represent the improvements from architectures. Did you, for these segmented, sorry, I got a question from Kimberly. For those segmented images, did you do all three phases as described prior, or is it deployed model? So we've done all three phases, and as I'll get to in the later part of the talk, the deployed model is actually available now. We have a web portal, deepcell.org, and we've also built plugins for different image analysis ecosystems, notably ImageJ and QPath. So the work to produce this model was, phase 1 and phase 2 of that process. And we've executed that already for this data set. If that wasn't clear, apologies. Yeah, so these are meant to compare, the improvements in sort of model development and model architecture, and they're quite noticeable. Here we've made a comparison between models that have been trained on other data sets, and so the FeatureNet models were actually this is the very first model that we developed for segmenting multiplex image tissues. It was trained on a data set specifically from MIBI, and it just does cell nuclei. And here you can see, it's quite poor compared to the models trained on the full data set. And we've also compared pre-trained models from CellPose developed by a group at Genelia Farms. This is a model that was designed to do whole cell segmentation, and as we see, the precision is quite good, but the recall is subpar, and the failure mode that's happening is that cells that don't have a good membrane label, this model misses. And so there's sort of two things that leads to improvements. There's sort of the model improvements, but the largest gain is actually from the data set improvements. And I actually suspect that if this model were trained on our data set, it would do fairly well. Although there are other reasons like why the multiplexed DeepCell method would likely be preferred. We've also, looked at the difference between, general models, so models trained on like the full TissueNet, and then models trained on subsets. There was some concern that, models trained on specialist models trained on subsets, might perform better. The reality is that we don't see that. Whether it's, training specialist models by platform or training specialist models by tissue. Roughly speaking, the generalist models are almost indistinguishable from the specialist models. And the reality is that, the general models are simply more valuable to the community and also easier to deploy. With these models, like you can actually, because you can perform whole cell segmentation, there are analyses that you can do that would be impossible otherwise. One of them is subcompartment analysis. And so we've here, looking at different markers, so Ki67, this is a marker that, is invariably, one would expect to be nuclear, while ECadherent is one that's expected to be in the membrane or cytoplasm. And, this is, both demonstrating that we can do these subcompartment analyses, but also as a sanity check. And so if you look at the, nuclear to cytoplasm ratio for these different markers, what you'll see is that the nuclear markers are nuclear, and the non-nuclear markers they're not nuclear. And this is sort of what you would expect. Great segmentations, how to 3D orientations, series Z-scan data. Yes. So 3 dimensions is in the not too distant future. If you take this model and then apply it to a slice in a 3-dimensional data set, it actually does perform quite well. This is not inherently a 3-dimensional model, though. And I think what's not appreciated is that an out-of-the-box solution like this, prior to this work, it did not exist for 2 dimensions, let alone 3. And so while we're optimistic that this would sort of create like a straightforward building block for building a performant 3-dimensional model, I think the reality is that the data sets aren't there, and the algorithm and the as far as what people are routinely producing. And honestly, the training data, prior to this work, it wasn't there. It wasn't there either. 2 dimensions was still an open problem before this work, and it made more sense to sort of solve a 2-dimensional problem before going off into 3 dimensions. Yeah, and I think this is the last bit I'll say on accuracy. So we have human annotations to we have sort of these inner annotator. We have annotations from humans that we can use as a comparison. And so if we, take our model and then ask, okay, how does the model annotator comparison compare with the inner annotator comparison? How do those stack up? This is shown in this plot here. And so in blue is sort of the human to human comparison. Red is the human to model comparison. And what we see is that, effectively these are indistinguishable statistically speaking. And so what we sort of infer from this is that the model is effectively at human level performance for whole cell segmentation. Not satisfied with just that analysis, though, we've actually taken the outputs of this model and had 4 independent pathologists do blinded comparisons. And so they're sort of given 2 example segmentations for a given data set, one produced by a human, another produced by our algorithm. And then they're just asked, which one is better, the one on the left, the one on the right. They don't know which one is the algorithm and they don't, or the human. And so when you look at that comparison, what falls out is what's on the right. And so a score of 1 means the human is better. A score of 5 means that the algorithm is better. The different annotators are colored by, are sort of given different colors. And so for these 3 different imaging data sets, again, the take home is that there's, the pathologists aren't able to demonstrate a preference for one or the other algorithm versus human. Okay, and so the last bit for this is, sorry, there's one more question. Perhaps about how do you combine information from multiple imaging channels? Okay, so I did not mention this. And so what the model expects is a two-channel image. One channel is the nucleus. The second channel is a membrane/cytoplasmic image. And so the reality is that people often use multiple markers for that. So it's not the case that there's one, pan-membrane marker or pan-cytoplasm marker that's available. Usually it's a smattering of them. And so what we've done is we've sort of just taken those and, for a given data set, and you just take the ones that look good and then you sum them together. And, what we've just observed empirically is that's enough to get us to this point where the models are fairly performant. There are use cases where you might want to use this different marker separately, and in that case transfer learning is something that you can do. You just annotate a relatively small data set for those use cases, and then fine tune this larger model. And I don't have slides to show. I don't have slides to show today, but we've done that in our collaboration with Mike Angelo, and it works fairly well. Yeah, so I think the last bit I want to say, and this is very preliminary stuff, but we're very excited about it, is that this whole cell segmentation model is going to enable morphological analyses of cells and tissues. And so here we've just gone through a comparison of looking at different morphological features. So here it's nuclear to cytoplasmic ratio. So top is what a high end-to-C ratio would look like versus a low. And this is then comparing, okay, what does a model produce versus what does the ground truth say? And you'll see that there's actually a very good correlation between the two, although there are some notable differences. So this is a density plot of the true versus predicted. The correlation, the R score is about 0.87. Yeah, and again, very early days, but we're starting to dig into what these sort of morphological analyses can tell us. And the two things that we're excited about is that one, it does look like there is cell type information encoded in morphology. And again, if you talk to any pathologist, that would not be surprising. But what's also exciting, so just to be clear, what we've done is we've sort of taken 4 different measures of morphology, so area, whole cell skewness, nucleocentricity, and then nuclear cytoplasmic ratio. And then we took that vector and then performed dimensionality reduction using UMAP, and then we plotted the UMAP and then colored some things. And so what's shown on the left is that you sort of get these 4 distinct clusters, and if you were to color by epithelial, epithelial versus not epithelial, you'll see sort of the epithelial cells predominantly live in different clusters, sort of indicating that there's some amount of cell type information in these morphological feature vectors. Sorry, there's a question. Will the recording be made available? Some of my colleagues should see this. Yes, this recording will be available. Yeah, so point 1 is, there's cell type information in morphology. Figuring out how to best extract that, we're still working on it. What's also exciting is that cell morphology is something that is preserved across all of these different imaging platforms. And what we think is that it would actually be a very nice and clever way of registering disparate data sets. And so this this shows this UMAP colored by these different platforms, and it shows that there's really no bias when you look at morphology. The CISIF, the MEBI, the IF, and the Vectra data sets, they're sort of distributed all throughout. Yeah. Okay, I think I'm running short on time, so I'm going to zip through some stuff. We love live cell imaging. The ability to get information about dynamics is very exciting. We're working on building a similar data set for live cell imaging, collecting nuclear cytoplasmic movies across commonly used cell lines and annotating them. It was a long effort, but we've just gotten to the point where getting entire movies annotated is scalable and affordable. And again, not just for cell nuclei, but also, for the entire cell. We've had a preprint where we sort of discuss what's possible with the nuclear annotations, and so we've developed deep learning models for doing both segmentation and tracking of cell nuclei, and being able to identify okay, our cells. If I look at cells across frames, are they the same cell, different cell, or is there a mother-daughter relationship? And being able to get that last point will let you assemble cell lineages. Okay, so these GIFs actually are working. So this is an example of our deep learning-based cell tracker. And so what you'll see is that as cells divide, they sort of get assigned different colors, which is sort of the model recognizing that they're no longer the same cell. And you sort of have to trust me that the lineage information is being recorded. What the pipeline currently produces is JSON that tells you which mothers are assigned to which daughters. Where this is heading is, being able to do whole cell segmentation and tracking in live cell imaging. So this is some very preliminary data that we produced where, with a deep learning model, we can do whole cell segmentation in label-free images, and then, having a cell tracking model sort of link things together in time. And what we're particularly excited about is, having a framework where we can actually annotate the edge cases, because that's where, sort of the, I personally think the exciting cell biology is, so there's, when you're sort of working in, when you're thinking about tracking models, there's a, notion that there are clean division events that go from, 1 cell into 2 cells. The reality is that, there's a lot of things that go wrong, and actually just having something flexible enough to catalog and identify and catalog what goes wrong, that's exciting. That's exciting to us. And so I think some examples of that are here. So on the bottom, you can sort of see 1 cell that divides into 3, and up top, you can sort of see examples of 1 cell dividing into 3, and then there's another where it sort of divides, changes its mind, gets back together, and then sort of redivides into 3, and sort of being able to reproduce these identity graphs. The data needs to be in a place where these edge cases are annotated, so you can actually do that. And we sort of have the infrastructure to do that now. which is one thing that we're working towards, which is a full stack AI infrastructure for doing all of these tasks. So compiling data, sort of tracking data across experiments, sending them off to be annotated, having the human in the loop infrastructure so that the annotation can be scalable and affordable, and sort of having all this done in a open and accessible manner for those of us in the academic community who work on nonprofit projects. And sort of what we envision is sort of having all this infrastructure in place, so the actual process of going from data to annotations and models is almost like turn the crank. And and so the marginal cost with this infrastructure, sort of what we envision is that the marginal cost of producing these data sets will go down because all you really need is the crowdsourcing labor, and then the labor of an expert to sort of oversee this process. And what we envision is that the labor cost for producing, say, like the next TissueNet, that would be on the order of $20,000 to $30,000, which might sound like a lot, but the reality is it's on the same scale as a moderate size sequencing experiment. And I think most people would agree, there's going to be much more utility out of these methods than what a single sequencing data set would give the community. Models and compute. I think I'm almost out of time. Spontaneous Russell, let me answer that question at the end, because I would like to get through this because everything I showed is great, but it's not valuable to the community if it just lives on our lab servers in Jupyter notebooks, and you need to have either a master's or a PhD in computer science to be able to access it. And so models and compute is actually like a very big area. Deep learning is great, but the accessibility is a big issue, and there's sort of two parts of it. There's a DevOps part, what's required to actually get the models to sort of work in your own hands. And then there's also the throughput part. So the reality is that biological images are larger in size, and that means the inference speeds are low. And this is a challenge for anyone who does either large-scale imaging experiments or wishes to make a computational pipeline widely available through the community. And I think the technical challenge is that because it's these pipelines that are valuable, there's a resource allocation problem. These pipelines have deep learning, and then they have other computational operations. The deep learning is best done on one set of computational infrastructure, e.g., GPUs. The other stuff is better done on other sets, and just having a single machine, sort of host entire pipelines, that's suboptimal. Ideally, what you want to be able to do is separate out these pipelines so that the deep learning operations take place on one set of compute nodes, and the other operations take place on other sets. If you're operating in the regime where data transfer is fast and cheap, that ends up being the cheaper and also the more scalable computational infrastructure. And so we've built software that does that in the cloud. So it's called DeepCell Kiosk. There's a preprint describing it on bioRxiv, and it's slated to appear in Nature Methods in a couple months. And effectively what this is it's a blast for cellular imaging. So just like how BLAST changed how people interact with sequencing data, you didn't need to get things operational locally anymore. You could just take your sequence, upload it to the BLAST server, and get the results back. The DeepCell Kiosk effectively performs the same function for cellular imaging. There's a computational infrastructure that's being hosted in the cloud. In this case, it's Google Cloud. It has this resource allocation problem solved already. So we've designed it so that the deep learning is accessed through a service, and the pipelines sort of access that deep learning by sending data to it and getting the results back, and they can do the rest of the computational operations happen on cheaper nodes. And we've also built this to be scalable. So if more data flows through it, i.e., if more people are trying to access the pipeline, the computational resources get scaled with the amount of data. And so if there's periods of high demand, the infrastructure grows. If things are low demand, the infrastructure shrinks. And this helps keep costs affordable, which is sort of what's needed to keep something like this operational. Because we have things hosted in the cloud, we can make plugins for different ecosystems. So for Fiji or QPath, and the way these plugins work is by taking data and then sending it to the cloud server and then getting the results back. What that means is that you can install it and not have to worry about, do I have CUDA installed or CUDNN or any of the prerequisites. None of that's required. You can just download the plugin and send it to an operational server. And so the version that's we have a instance of this operational DeepCell.org. It hosts models that we think are useful. One of the models that's currently being hosted is this multiplexed imaging model. And there's example data that you can download and try. And these plugins are also available as well. We've done some pretty thorough benchmarking to make sure that this is actually is scalable. And we've also benchmarked like how much it costs. If you're the rule of thumb is that, for 1 million images, this infrastructure can process that in about 5 hours, and it costs in the order of $100 to $200, depending on the actual details of the configuration of the cluster. And what's nice about this is that we can actually build pipelines that make use of multiple deep learning models. And so pipelines that say, for instance, do nuclear segmentation and then cell tracking. This is all supported through our ecosystem. And again, even those are accessible through the web portal plugin, or also a command line interface if you're sort of more programming savvy. We actually started hosting tutorials showing folks how to use this. And so this is something that I was really, sort of made my mouth drop because, we hosted a tutorial at HHMI's Image to Knowledge seminar, and some folks from the La Jolla Imaging, the La Jolla Imaging Corps, I think their Twitter handle is here, they saw what we did, and they, sort of put their own data through this pipeline. And so this is, what they posted on Twitter. Their data, I believe it's like human tonsil. They have nuclear, and then also I believe it's either CD4 or CD45 as their membrane marker. And this is all done in QPath. None of it required any, anything else, just QPath and the requisite plugin. And here they've got their cell segmentations in yellow, and they can go do all the downstream analyses in QPath as they wish. So all of this is open source software, and so there's stuff that we've developed that was essential for this work that I didn't get a chance to talk about. All of it's available. So there's DeepCell Label. To actually do the crowdsourcing, we had to write our own annotation software because what existed wasn't adequate. And so this is what enabled us to do all the annotation that you saw saw in this talk today. And then DeepCell data sets host some of the annotated training data that we've developed for developing these single cell analysis models. The multiplexed imaging data set is not there because we're still in the midst of preparing this manuscript. The models development, we have a repository, DeepCell TF, and so these are implementations of deep learning models for single cell analysis. All the models that we sort of showed today, we use this repository to train those models. And then lastly, there's the DeepCell Kiosk, and so if you're interested in the cloud computing and sort of hosting your own collection of models and doing scalable inference, that's also available as an open source package. And then there are DeepCell applications, pre-trained models for single cell analysis. And so we sort of envision this ecosystem as sort of attempts at solutions for the data challenges, the model challenges, and the inference challenges involved in getting software 2.0 to push this single cell biology and also spatial genomics forward. And so everything's available at our lab GitHub. If you like the packages, I say please add a star to the GitHub repositories. It's one metric that funding agencies use to determine, which packages are being used and which ones are not. And that's important for making sure that this work continues. And again, if you want to try it, things are operational at deepcell.org. The plugins can be downloaded, and a detailed tutorial, sort of showing, sort of the ins and outs of this ecosystem, be it the labeling software, the pre-trained models, how to install the plugins, all of that. That's available at this Intro to DeepCell, which we put together for this HMI tutorial. And that is it. So I need to give a big thanks to all the folks in my lab. Obviously, this is a huge collection of work. There's no way that this would have happened without them. And a lot of this stuff has been sort of been a group effort. But I would like to single some folks out. William Graff is our software engineer, and he's sort of been the person who has made it possible for us to go beyond academic software and to create create software that has the requisite software engineering that these efforts require. Eric Moen is a postdoctoral fellow in the lab, and he's been the one who sort of innovated this human loop approach, which is what made all the crowdsourced annotation viable. The TissueNet has been a collaboration between my lab and Michelangelo's lab, and we've both been equal partners. Noah Greenwald has been a graduate student in Mike's lab who has sort of pushed the tissue segmentation forward, and Geneva Miller has been the counterpart in my lab who sort of fleshed out the software engineering required to help make that happen. And then lastly, Tom Doherty is a software engineer in my lab. Morgan Schwartz has been essential in a variety of programming tasks. Dylan Bannon, he's the graduate student who spearheaded our efforts in cloud computing, and the reason his work is the reason why we can have the sort of centralized model for deploying models to VG, ImageNet, and the whole community. Lastly, I need to thank some additional collaborators. Liat Karen, Wong Kai, Yisong Yu, and then also Eric Osterman at Cloud Posse. And then lastly, I need to thank the folks who help support this work. The NIH through a sub award, the Paul Allen Family Foundation through through centers that they funded at Stanford and also at Caltech and University of Washington, and then also Shirley K. Kersey Foundation, the Reed Allen Foundation, variety of partners, and then also a variety of centers at Caltech who've contributed in one way or another. And with that, I will stop and leave, stay around for questions.
Thank you, David. This was a very impressive segmentation. Pioneering talk, I would say. One of the state of art already. Any questions, guys?
So I see quite a few in a chat box, but some probably have already been addressed. But Ahmet, I don't know if you want to raise some questions. Sure. Or
I think the last one, that one was missed.
The last one was missed. Assumption or combination for tissue section thickness in the live cell model. So that sort of goes into the curation process because we compile data sets from a large number of labs. And generally speaking, it sort of needs to be thin enough where you can identify single cells within like that cross section. If there are too many cases of cells on top of cells because you captured too thick, then we generally speaking didn't use it.
Okay, I was just curious about that to see off the bat if your section is too thick, was it going to mess up the model? So
I would say it probably would. There's going to be a limit where if it's too thick where it stops working. For those cases, three-dimensional imaging is probably like what's best anyway. And what we envision doing in the future, in the not too distant future, is using this model as like a starting point for creating a three-dimensional data set. And so what you'd imagine is that you take a selection of Z slices, use this model as a starting point, creating segmentations for each slice, and then the annotation task then becomes, connecting things across slices as opposed to creating annotations from scratch. And that I think would go fairly quickly, but it would be a fair body of work. And I think one thing that's sort of standing in the way in that is just making sure that what we've done already gets funded and gets supported and funded. Before this work, the comments that I've heard from people was that this was not possible, that crowdsourcing can't work on data like this, and that humans can't perform segmentation on data like this, and that it's too hard. And so I think we've sort of demonstrated concretely that that's not the case. And I think for, for two-dimensional tissue segmentation, I don't want to say this problem is solved, but if I were to, sort of put the DeepMind, hype hat on, if we're going to call 90% solution solved, then for most intents and purposes, I'd say like we have like a pretty effective solution for this. We need to get some amount of support to make sure this continues. And, if that happened, one thing we'd like to do is 3 dimensions. We don't think it would take us that long to develop a model to do that, but like it is like a non-trivial engineering effort.
Yeah, I have kind of 2 quick questions. One was related to what GC was about to ask as well. And so when you do all your segmentation, you use, they're really kind of extremely high-plex data, right? So how did you kind of combine different proteins or genes in this high-dimensional data to do your segmentation, or you just pick specific markers to do segmentation and then look at the expression pattern of other?
Yeah, so I think the ideal, the solution would be like a joint like wet lab / computational effort. And so what's needed on the what would be ideal on the wet lab would be if there was some like magical panel of antibodies where if you just added it to like any experiment on any tissue, you would be guaranteed to get good membrane signal and staining. The reality is that marker panel doesn't exist. So Noah was actually trying to develop this, but COVID happened, and the reality is like that experimental work, sort of slowed down. But you can't let perfect be the enemy of the good. And so what we decided to do instead was say like, okay, well, if the data sets that we have, let's just identify, good markers. So there's some markers that we know are going to be good, right? So for instance, like ECAT here is going to sustain, in a membrane-like pattern for epithelial tissues. For immune cells, CD45 is going to be, sort of membrane staining as well. The curation is effectively, done manually in that sense at the beginning stage, and it's just, of the data sets that people were gracious enough to contribute to this effort, which markers, had sufficient membrane contrast to warrant training on, warrant annotation and then training on.
Okay. Yeah. Thanks. I see Guochen unmuted himself. Maybe you want to further add something?
Yeah, I have a different question. So David, so you mentioned at the beginning there's one of the challenge is to scale it up. How do you do this in the large scale data? And if I understand correctly, the approach that you're using right now is pretty much using the crowd sourcing, right? So do you think that's the model for everyone? That's the future, or that's something that should be done in different models?
Yeah, so I think it depends on the task, right? So I mean, this is the reason why I said like, hey, you're gonna love this talk is because like you've done like some really remarkable stuff on sort of modeling the spatial transcriptomics data, and doing the whole cell segmentation has been like a big barrier to going from like, hey, raw data to here's what the software packages that you've developed need. I think like the reality is that like a big part of like that challenge is I don't want to say like it's entirely solved, but we're 90% of the way there. This model of crowdsourcing. So there are other challenges, right? So like another challenge would be like spot detection. And so if you're doing spot detection in tissues, autofluorescence makes it like a non-trivial effort. That's not something that you can crowdsource away. And the reality is just that there are just too many spots in tissues to be able to, say like, hey, like we've curated every single one, right? Like I don't view that as being like feasible. And so you have to, there are other things that like you'll, that one I think should or could do. But for tasks like this, I think crowdsourcing is the solution. And I think the way in which we've done crowdsourcing is a solution. And there's sort of two things, and like all things, like the devil's are in the details. And I think there are two things that about what we've done that that made it work. One is the human in the loop approach. And this is, what conceptually has ended up happening is that the models end up being a vehicle for delivering annotator insights to expert insights to annotators. And in that way, the annotators don't have to start learning how to do this task from scratch. They actually have like a pretty good starting point for what they should be doing. And it's possible to guide them like throughout the process, which gets to the second thing. The way in which we do crowdsourcing is not the wild, wild west mechanical Turk way of doing things. We've actually partnered with, managed crowds so that we have access to workers for, sort of guaranteed numbers of hours, and also some amount of management so that we can deliver feedback about what's working and what's not working. And so the role of the experts in this process, at the beginning, it's, creating the pilot annotations, but the bulk of their time is actually spent, doing some quality control and then delivering feedback. So it's not that experts don't have to do anything where they, sit back, like this, and the crowd, solves their problems for them. But the flavor of the work is actually, what experts tend to tolerate. It's supervisory in nature, less, rote mechanical work. And so I think for a lot of tasks, this approach is going to be what gets our field the data it needs to sort of get the performant machine learning models that are necessary for this highly multiplex single cell future that we're all envisioning. So the reality is, like, without it, like every manuscript takes inordinate amounts of like manual labor to just get to the single cell resolution. Like everyone's doing it. It's just that no one's talking about like those sacrifices. And I think with automating those steps, it's actually going to accelerate like the rate of discovery because those students won't have to perform those steps. I think Mike Angelo, who's also very active in this multiplexed imaging space, and they do a fair amount of image analysis. He sort of asked me like what the impact of this has been on his lab, and like his quote was, things that used to take a month now take on the order of a couple hours, and most of that time is spent, sort of identifying like which markers should we send through this algorithm. I think this could be the future for everybody. And for cases where the data doesn't exist, what we've outlined is the sensible way to get there. When you look at the number of hours required to create a TissueNet, it was on the order, I want to say around 3,000 or 4,000 annotator hours, a much smaller fraction for expert hours. And when you sort of take that and multiply it by, okay, what's the hourly rate for a crowd worker? Like a sensible rate is around, $5 to $6 an hour. And that means, the next TissueNet, it's not unreasonable to think that would cost us around $20,000, which might sound like a big sticker price, but the reality is that even for academic budgets, for something that's this useful, it's a small price. And honestly, like that's what I like the most about what we've done, and I'd say that's what I like the most about what my postdoc Eric Moen has done is much of the work has been in figuring out what to do to get that marginal cost of data annotation down, so that these projects can just be stamped out over the next couple of years.
It's very interesting model.
Yeah.
So David, I know we're running late, but I do have a.
I'll stick around. I've got my next meeting's not till 1, so.
That's great. I do have another kind of quick question. So, have you done something like a standard tissue morphology image, standard tissue histology image, like H&AE? Maybe I don't know much about tissue histology. That would be easier for you to do segmentation,
So for nuclear segmentation, yes. There's a really nice data set produced by folks at University of Warwick. I think it's called PanNuc. And what they did is they annotated cell nuclei and then also had pathologists assign some notion of cell type. And so you could distinguish like inflammatory cells from cancer cells, from epithelial cells or whatnot. And we've played around with like those data sets. I think the challenge is, for H&E images currently, what's going to be the ground truth? And if it's human generated ground truth, that has errors. How well can a pathologist distinguish a cancer cell from an immune cell? How well can they distinguish a cancer cell from an epithelial cell? Those are, I think those are issues. One thing that we've been thinking about doing is sort of collecting registered data sets where you have H&E images and then also these multiplexed images as well. And so all the insights that you get from multiplexed imaging, can you sort of use that to create a ground truth for the H&E images, and then train models that can pull that out of H&E images. That's one thing that we thought about, but it's just getting this stuff sort of done with the requisite amount of polish has been like a more than full-time job. One thing that sort of gives me some pause about doing that is, I sort of, just like there's, sort of software of the future and legacy software, I think there's data of the future and legacy data. And although there's a lot of really nice data sets with compiled H&E images and, clinical information, I think the reality is that H&E images are legacy data. And so how much time to devote to, extracting information from legacy data sets, I think that's not clear. And the reality is that, time and resources are limited. And we've opted to sort of spend our time sort of more on the data sets that we think are going to be, what the future insights are built off of. Yeah.
Yep. But even in the future, I think, for example, 10x genomics, right? They have no capability to put the H and the E and spatial sequencing together. That's why I'm asking. If you are able to do both, that would be transformative.
Yeah, so we can do both. And the reality is if like the NIH called me tomorrow and said, Dave, here's like a UO1 with like, $1 million a year, like I could promise I could like do that like within like the next like, 6 months or something. The reality is that's not where we are right now. And so like our resources are limited, and so we can't do everything. So we sort of had to like pick what to do first. And this what we presented today, I think that was the right decision. Like a solution for whole cell segmentation. Everybody's needed this for a number of years, and I think we have a pretty compelling solution.
Well, I think there's one more question in the chat from Valen.
Yes. Can the model output surface level cell phenotypes using the cell morphology, or is that a secondary step? That's a secondary step. So what the model spits out is a label image where the pixels belonging to a particular cell are given a unique integer ID. And then once you have, and then you also get a similar label image for the cell nuclei. Once you have those two label images, all the morphology stuff is secondary post-processing.
Yeah, I have a quick question from this side before we
Go for it.
Too late, but so the spot detection, what is the highest accuracy that you get so far?
I can't tell you because we're in the midst of figuring out how to do that well. I would say, like our first attempts were on data sets where we had just sort of manually fine-tuned some of the classical algorithms, and we got and so the idea is that if you sort of fine-tune on enough data sets, the model will learn how to do the task, and you won't have to fine-tune, and that's sort of like the value add. You won't be more accurate than the classical methods, but it'll be easier to use. And so if you have like an image, you can just run it through, and then you get a result. And I think if my memory serves me correct, we're sort of using the F1 score comparing like the ground truth and what the model produced, and that was around like an F1 score of like 0.8-ish. that's not really a measure of like how well you're doing. That's a measure of how much does the deep learning model agree with like their classical produced annotation. As we sort of, progressed, we wanted to do certain things. One thing we wanted was some subpixel accuracy because, for the multiplex methods, registering across the images, that actually matters. And so getting the deep learning to be able to do that, that was a little bit of an exploration. We're not the only method, we're not the only folks who are interested in this. There's a paper, Deep Storm, and I believe there's another Anapalm, where they show like deep learning can actually get the subpixel resolution using inference. Figuring out how to do that, given the data that we were working with, though, that was I think that was something that we had to figure out. And like there's some machine learning type insights into what the loss function needs to look like to be able to do that. So we can do that. The real thing that bothers I would say bothers us, that bothers me, is treating this one classical algorithm as like the origin of your ground truth, and that doesn't feel right. And so we spent some time thinking about, are there other better things to do? And so what we've settled on, and so this is work that a former postdoc, Nitzan Razin, started working on, and a graduate student, Emily Lauscher, has carried on, is, okay, let's say like, we can't get one algorithm. We don't have a human generated ground truth. And we don't necessarily want to trust one algorithm. What else can we do? And sort of what they sort of decided on is like, okay, well, as opposed to using one algorithm, how about we use a collection of algorithms? And so there are a collection of algorithms for doing spot detection that vary, Laplacian and Gaussian, and whatnot. And so what we've been working on is like, okay, let's annotate some data with these collection of algorithms. So each one is going to produce some prediction of where spots are. Let's use generative modeling to sort of identify, okay, what are, consensus spots, areas of agreement, which we can, which the models can, then agree, like, hey, these are likely real spots, and also what are areas of disagreement, and so these may or may not be, false positives. And then with that information, use that to train deep learning models to do the spot detection, training on the output of this generative modeling process, as opposed to any individual classical algorithm. Also, by the way, like this isn't something that we thought of. This is an approach developed by some folks at Stanford. I'm blanking on the PI's name, but the name of the method that first started doing this is called Snorkel, and it's a variant of weakly supervised learning. And we're just now starting to get that to work, and so getting the generative modeling to produce results that make sense. And right now we're getting the deep learning to produce results on that generative modeling to make sense. And realize it's too early for me to sit here and say like, oh yeah, like we have F1 score of 0.9. And everything's great, and we'll solve spot detection. Nature papers coming out in 3 months. I can't do that because like that's not reality. We have to see how well things are. We have to make sure that we're being fair and unbiased as far as like how we're benchmarking things. We need to talk to folks we work with, like Long Kai's lab, to see like, okay, we've developed this method. How useful is it? Are the results accurate enough where you can say like, okay, I don't need to fine tune anymore, or are there still lingering issues? And then once we have that assessment, I'll be able to go in front of the community and say like, okay, well, here's how well it performs. Before then it's just hype, and I think the artificial intelligence field has enough issues with hype, and they don't need one more person adding to it.
Yeah, good saying. Thank you so much.
Yeah.
Any other questions before we wrap up today? Okay, let's thank David again. I think there's a clap tool here. I want to find it again. Reactions, I think. Oh, but this is not okay. Let's clap again, physically. Thank you so much. And
thank you for having me. It's a pleasure.
Thank you so much. I appreciate it. And next week we will be continuing with Dr. Emma Lundberg for Spatial Proteomics talk. And as you know, they've been running this pretty cool Cell Atlas project, right? The website Protein Atlas as well. And also she's been at Stanford, and she kind of transferred codecs to also Sweden. So she's going to be probably recently, hopefully showing some more multiplex data in that talk as well. And that will be next Friday at 2 p.m. And then we're ready to end the talk and today's session, and hope to see you next week, and have a nice weekend. Thank you, guys.
Thank you, everyone.